Okay, so just one announcement about next week. You have your third exam, and this will be the last exam before the final. This exam is going to cover cell communication, photosynthesis, cellular respiration, and fermentation. Cell division, cancer, and sexual reproduction and meiosis. Okay, so again, cell communication, photosynthesis, cellular respiration, and fermentation, cell division, cancer, and meiosis will be on the exam next week. And it'll be the same format as the other two. So half the points on the exam are going to come from multiple choice questions, and the other half will come from answers that can include short answer, fill in the blank, labeling if appropriate. Okay, so we're going to continue on. We're in the middle of sexual reproduction and meiosis, and we left off the last class by watching the animation that described the different stages of meiosis and what happens. And just to remind you, in meiosis, meiosis involves two cell divisions. So you're going to start off with a diploid cell. In the first division, we call meiosis 1. And that's going to produce two daughter cells that are both haploid. So you half the number of chromosomes in the first division. And then the cells, both cells are going to undergo meiosis 2. And at the end of meiosis 2, you're going to end up with 4 daughter cells. And all of them are haploid. Okay, so ultimately in meiosis, you go from a diploid cell to producing 4 haploid daughter cells. And so you can see that the daughter cells have half the number of chromosomes as the parent cell, so they're not genetically identical to the parent cell. They're also not genetically identical to each other, and we'll see the processes that lead to that. Okay, also then, in the animation that we watched last week, we broke meiosis 1 and meiosis 2 down each into 5 stages. So meiosis 1 is simply broken down into prophase 1, pro-metaphase 1, metaphase 1, anaphase 1, and telophase 1. And then meiosis 2 is broken down into the same stages, and you just put a 2 after each of the stages. So you have prophase 2, pro-metaphase 2, metaphase 2, anaphase 2, and telophase 2. Okay, and so what we're going to do now is discuss what happens in a certain number of the, we're not going to discuss every stage, but we will discuss some important things that happen during meiosis, during some of the stages. And so similar to what we did for mitosis, for meiosis I have a slide for each phase of meiosis. And so just one thing that we're skipping over is there's also an interphase that precedes meiosis. And during that interphase, one thing you're obviously going to do is you're going to replicate the DNA. And similar to mitosis, you only replicate the DNA once. Okay, so before meiosis, you're going to replicate the DNA once during interphase, and then following replicating it once, it's going to be followed by 2 cell divisions in meiosis. Okay, so this slide should be labeled prophase 1. So this is the first phase, and there's a lot of important, there's a lot of important things that happen in this stage, so it's important that you, this will be a very important stage. So one thing that's going to happen is the chromosomes condense. So similar to mitosis, the chromosomes are going to become very compact during this process, so chromosomes are going to condense. Synapsis occurs. So this is a really important event. And synapsis is the pairing and physical connection, is the pairing of homologous chromosomes and their physical connection to each other. Okay, so synapsis is the process of the homologous chromosomes pairing up and becoming physically connected. Okay, so again, synapsis is the process of the homologous chromosomes pairing up and becoming physically connected. So in your cells, the 2 chromosome number 1s would pair up and become physically connected. The 2 chromosome 2s are going to pair up, etc. All the homologous chromosomes are going to pair up during synapsis. And then after synapsis, you're going to have crossing over occurring. And crossing over is an event where you get the exchange of DNA between 2 non-sister chromatids. Okay, so once the homologous chromosomes, each which consists of a pair of sister chromatids, pair up next to each other, they're going to exchange segments of DNA between non-sister chromatids, and that's what happens in crossing over. And this is one event that leads to the daughter cells being different from each other. It also leads to genetic variation and sexual reproduction. Okay, so one, it's going to lead to the daughter cells being genetically unique, and also it's going to lead to genetic variation in the process of sexual reproduction. Okay, also then your nuclear envelope, and I'll show you a picture showing synapsis and crossing over in a second, and we'll also watch an animation that covers crossing over. Your nuclear envelope is going to break down. And the spindle begins to form. Okay, so all that's going to happen in prophase 1. Okay, the really 2 important ones that you want to remember are synapsis and crossing over. And those are 2 things that occur in meiosis that do not occur in mitosis. So synapsis and crossing over do not happen in mitosis. Okay, so this is showing you the process of synapsis and then crossing over. So these 2 are homologous chromosomes, and what they're showing you is the pairing of the 2 and then how they become physically connected. And then once they're physically connected with each other, 2 non-sister chromatids can exchange DNA. That exchange of DNA is crossing over, and then after that, you're going to proceed through the phase. Okay, so you have pairing of the homologous chromosomes first, which is synapsis, and then following that, you can have crossing over. In terms of crossing over, this kind of shows you how crossing over is going to lead to the daughter cells being genetically unique from each other. So this is showing you a crossing over event that occurs between these 2 non-sister chromatids. And then once crossing over occurs, you can see that these 2 sister chromatids are no longer identical to each other, because this chromatid here has a piece of the pink chromatid attached to it rather than being one long blue chromatid. And then when you separate those sister chromatids, this cell is going to be different from that cell. And then the same thing happens down here. When you split those sister chromatids, that's going to make that cell different from that cell. Okay, so crossing over is one thing that's going to lead to the daughter cells being genetically unique from each other. Also, this will show you how crossing over will lead to an increase in genetic variation that occurs during sexual reproduction. And then I'll explain it further once we watch the video.
Crossing over during meiosis allows recombination of genes between homologous chromosomes. This alters the linkage between genes on the same chromosome. Crossing over occurs frequently during prophase 1 of meiosis, when homologous chromosomes associate with each other in a structure called a tetrad. First, consider the possible inheritance of alleles for 2 genes on a particular chromosome when crossing over does not occur. Recall that one member of each pair of homologous chromosomes came from one parent. The other member came from the other parent. In this example, one parental chromosome had alleles capital A and capital B, while the other had alleles lowercase a and lowercase b. Without crossing over, each haploid cell produced will inherit a chromosome with the same combination of alleles as was on one of the parental chromosomes. Thus, the arrangement of linked genes is not altered in this case. Now, consider the case where there is crossing over in the region between the 2 genes. Since the crossing over is only between 2 individual chromatids, the possible haploid cells after meiosis have 4 different genetic combinations. 2 will come from the chromatids that did not participate in crossing over and thus will still have the parental combination of alleles, either capital A and capital B, or lowercase a and lowercase b. The other 2 will be recombinant, with either capital A and lowercase b, or lowercase a and capital B alleles. Thus, crossing over allows for the reassortment of linked genes.
Okay, so in terms of talking about genetic variation, the point that the animation made was that if crossing over does not occur, then the only combination of genes that you could pass on to offspring would be a capital A with a capital B, or a lowercase a with a lowercase b. You would only have those 2 options for passing if crossing over didn't occur. But if crossing over occurs, now you can produce an offspring that carries capital A with lowercase b, and that's a new combination. And then you can also produce an offspring that has lowercase a with capital B. That's a different combination. So crossing over leads to an increase in genetic variation in the next generation because it's going to basically mix up the alleles on a chromosome. Okay. So again, the crossing over that's happening again during prophase 1. This is pro-metaphase 1, so all you have to do is label it. I'm not going to have you remember anything specific about this phase, but this is pro-metaphase 1. And then after pro-metaphase 1, you're going to have metaphase 1 occurring. And it's important for metaphase 1 to know what's aligning down the center of the cell. And in metaphase 1, it's pairs of homologous chromosomes that are aligning down the center of the cell. So that's different from mitosis. So in mitosis, it was chromosomes that align down the center of the cell. In metaphase 1, it's pairs of homologous chromosomes align down the center of the cell. Okay, so one thing that we kind of skipped over were the chromosomes becoming attached to the spindle. We skipped over that, but it is important for you to know how they're attached to the spindle once they're at this point. So remember in mitosis, what happens, I'm going to draw mitosis over here. You have a chromosome, and then this chromosome is going to be attached to microtubules coming from both centrosomes. So it's attached to both poles. That's mitosis. Okay? In metaphase 1 of meiosis 1, you're going to have pairs of homologous chromosomes lining up. So in my drawing, here's one pair, here's the second pair. And in this case, each chromosome is only attached to one pole. So this pole has microtubules that attach to this chromosome and microtubules that attach to this chromosome. And then the other pole has microtubules that will attach to the other chromosome in the pair, and then the same thing down here. Okay, so each chromosome is only attached to one pole. And so what's going to happen later on is that when you have separation during anaphase 1, you're going to separate homologous chromosomes from each other, not sister chromatids. Okay, so in the first anaphase, you're not going to separate sister chromatids from each other. You're separating homologous chromosomes. Okay, and then it's important to note that one thing that happens because of how things align down the center of the cell that's important in terms of meiosis 1 is independent assortment. Okay, so independent assortment is something that's going to happen because of the way these pairs of homologous chromosomes align down the center of the cell. So here they're aligning, and then you actually do the sorting process in anaphase 1, but independent assortment comes about because of the way that they align. Okay, and that happens because how each pair is orientated when it lines up is random. Okay, so let me change the color of one of these. Okay, so let's say that the red chromosomes are the one that you inherited from mom, and the black chromosomes are the one that you inherited from dad. Okay, so again, red from mom, black from dad. So if we start with this pair up here, if the pair lines up like this, then the black chromosome is going to go to this cell, and the red chromosome is going to go to that cell. But if it lines up the opposite way, if they're flipped, then the black goes here, and the red goes to the other cell. Okay? Now, whether this pair lines up like this or is flipped the other way, that's just a 50-50 chance. Okay? So it has a 50% chance of lining up like this and a 50% chance of lining up the other way around. It's random. It's like flipping a coin. Okay? And then how this pair aligns is completely independent of how this pair aligns, which means that if this pair lines with the red over here and the black over here, that does not mean that this one also has to line up red over here, black over here. This could line up either this way or the opposite way, and this pair has a 50-50 chance of lining up either way, and it happens independently of this pair. Okay, so because of that, when you sort them, each pair is going to be sorted independent of the other pairs. Okay? And what that means is that each time you have a meiosis, in a cell in your body, then you're going to have basically the chance for a different alignment occurring, and each different alignment can lead to different gametes. Okay? So meaning that, let's say things line up like this, you end up with one daughter cell at the end of meiosis that has 2 black chromosomes and one that has 2 red. But if this one pair here is flipped, that's going to produce a new product. Now you have one cell that has a black and a red, and then another cell that has a black and a red. Okay, so there's different ways that things can align down the center of the cell, and each different alignment is going to produce a different outcome. Okay, and so independent assortment is another thing that leads to the leads to the daughter cells being genetically unique, and it also increases genetic variation in the next generation. Okay, so independent assortment is the second thing that occurs, that one leads to the daughter cells being genetically unique, and then also increases genetic variation in sexually reproducing organisms. Okay? Now, in terms of the number of different ways that things could align, that's something you can actually calculate, and they introduce this to you in the lab. So in terms of calculating the number of different alignments that can occur during metaphase 1, you use the equation 2 to the nth power. Okay, so that's the equation for calculating the number of different alignments during metaphase 1. And in this equation, n is equal to the number of pairs of chromosomes. Okay, so for example, for the cell on the slide, there's 3 pairs. So the number of different alignments is 2 to the 3rd power, which is 8. Okay, so basically there's 8 different ways that the chromosomes could align down the center of the cell, and there's 8 different ways they're going to lead to different gametes being produced. Okay, Now, in terms of humans, we have 23 pairs, which means that the number of different ways that things can align in our cells is 2 to the 23rd power, which is almost 8.4 million different ways. So in your cells, there's 8.4 million different ways that things could align down the center of the cell, and that's going to increase, obviously, genetic variation in the next generation. And then you add on top of that the crossing over that occurred in prophase 1, and that increases it even more. Okay, do you guys have any questions about independent assortment? Okay, so, and also keep in mind that independent assortment and crossing over are only going to occur in meiosis 1. Those two things do not occur in meiosis 2. Okay, and so this is just showing you again in a picture fashion how different alignments can lead to different products. So this is showing you a cell that has 3 pairs of chromosomes, and it's showing you 4 different alternatives. So there's 8 total, but it's showing you 4 alternatives for how things could align, and each different alignment leads to different gametes being produced as a result of that division process. Okay. And then just to add on top of that, we'll watch an animation which basically describes the same thing.
During metaphase 1 of meiosis, each joined pair of homologs lines up on the metaphase plate. The orientation of each pair on the spindle axis is random. Either the maternal or paternal homolog may orient toward a given pole. The number of possible chromosome orientations equals 2 raised to the power of the number of chromosome pairs. In this hypothetical cell with 3 chromosome pairs, 8 2 to the 3rd possible orientations exist. Each orientation produces gametes with different combinations of parental chromosomes.
Okay. So again, that's kind of alluding to independent assortment and how it will increase genetic variation. Okay, so the next phase after metaphase 1, so remember we skipped, there's prophase, prometaphase 1, metaphase 1, and then this half of the slide is anaphase 1. And for anaphase 1, you just want to know what's being separated and moving towards opposite poles, and the answer is homologous chromosomes. Okay, so in anaphase 1, homologous chromosomes separate from each other. And move to opposite poles. Okay, so homologous chromosomes are going to separate from each other during this phase and then move to opposite poles. So what's happening in this case is you started off with a cell that had 6 chromosomes, and what happens during this is once these separate from each other, you only have 3 chromosomes going over here and 3 chromosomes going over here. So at the end of this phase, or at the end of this first division, you're going to produce 2 daughter cells that each only have 3 chromosomes. So you half the number of chromosomes in that first division. Okay, so then on the right-hand side of the slide, you just need to label it. So then anaphase 1 is going to be followed by a telophase 1. And then telophase 1 is going to be followed by a cytokinesis. And at the end, what you're going to have is 2 daughter cells. Okay, so this is what you have after telophase 1 and cytokinesis. You have 2 daughter cells, each with half the number of chromosomes as the parent cell. But remember that each chromosome in these cells still consists of a pair of sister chromatids because you haven't separated them yet. Okay, and so then you just go down the line again. So this is prophase 2, nothing that you need to remember from that one. This is prometaphase 2, again, nothing that I want you to remember from this phase. And then this is metaphase 2. And you do need to know, in this case, what aligns down the center of the cell. And then we skipped over how they become attached, but you should know how they are attached. So first, it's chromosomes that align down the center of the cell in this metaphase 2. So in metaphase 2, chromosomes align down the center of the cell. And in this case, you have something, it looks the same as mitosis because each chromosome is attached to both poles. So this red chromosome here is attached to both poles, so is this blue one, and so is this blue one down here. Okay, so then the next phase you have is anaphase 2. And in anaphase 2, you're going to separate the sister chromatids from each other, and the sister chromatids are going to move to opposite poles. And then that's followed by a telophase 2 and a cytokinesis. And then you're left with 4 haploid daughter cells, which again are going to have half the number of chromosomes as the cell you started with. So each of these cells has 3 when we started with a cell that had 6. And you can see that each daughter cell is unique from the other ones in terms of their DNA content, and they're also going to be genetically unique from the parent cell that they came from. Okay, do you guys have any questions about what's going to happen in meiosis or any of the important events? Okay, so we've mentioned 2 things that occur during meiosis that increase genetic variation in sexually reproducing organisms. So in sexually reproducing organisms, 2 things that lead to genetic variation in the next generation are going to be crossing over in independent assortment. Those both occur during meiosis. And then a third thing that occurs, but this does not occur during meiosis, is random fertilization. Okay, meaning that one individual can produce a bunch of different types of gametes. The second individual that's going to be involved in the reproduction process can also produce a bunch of different types of gametes, but the 2 gametes that come together in fertilization is random. So meaning that you don't have the same 2 gametes coming together every time, so that's each time you have a fertilization event, it's most likely that the 2 gametes coming together are going to be different than the other fertilization events that occur. Okay, so these are 3 things that occur in a sexual life cycle that are going to increase genetic variation in the next generation. Okay, so this is just showing you in picture format. This is just useful for studying a comparison of meiosis with mitosis. So meiosis is on the left and mitosis is on the right. So you can see that one of the differences in meiosis you have 2 cell divisions, whereas in mitosis you only have 1. And then in terms of the daughter cells you produce, in meiosis you're going to produce 4 haploid daughter cells, so 4 cells that have half the number of chromosomes as the starting cell. And in mitosis you only produce 2 daughter cells, and the 2 daughter cells have the same number of chromosomes as the parent cell. And then in addition to that, the 4 haploid daughter cells you produce here are genetically unique from each other, so they're not genetically identical, and they're also not genetically identical to the parent cell. But over here, these 2 cells for mitosis, the 2 daughter cells of mitosis, are genetically identical to each other, and they're genetically identical to the parent cell that they came from. And then 2 things that aren't shown on the slide that are going to happen during meiosis that don't happen in mitosis, well, we'll say 3, is synapsis, crossing over, and independent assortment. Those all occur during meiosis, but do not occur during mitosis. Okay, so crossing over can rarely occur during mitosis, but we're just going to say that it doesn't happen. It can rarely occur, but synapsis and and independent assortment certainly do not happen during mitosis. Okay, so that's just comparing mitosis with meiosis because we've talked about both now. This is just taken from your book, and what I'm going to do is convert this table into the language that I used when I was describing so some of it's going to be changed using the language that I used when I was describing the phases, so you might want to change it as I go through this. But this is comparing mitosis with what happens in meiosis 1 versus what happens in meiosis 2. So the first question that they're asking is, does synapsis occur during prophase? And for mitosis, it's a no. For meiosis 1, it's yes, and specifically it happens in prophase 1. And then for meiosis 2, again, it's no. Crossing over during prophase, mitosis, we're just going to put no down. Meiosis 1, we put yes. And then meiosis 2, again, we're going to put no down. Okay. Also, it's important to know how the chromosomes are attached to the poles. In mitosis, each chromosome is attached to both poles. Okay. In meiosis 1, each chromosome is only attached to one pole. And then in meiosis 2, each chromosome is again going to be attached to both poles. Okay. And then it's important to know what's aligning down the center of the cell in metaphase. In mitosis, we said chromosomes. Okay, so chromosomes are going to line down the center of the cell. In meiosis 1, it's pairs of homologous chromosomes. So for meiosis 1, it's pairs of homologous chromosomes that are lining down the center of the cell. And then for meiosis 2, it's chromosomes again. Okay, so for meiosis 2, you have chromosomes lining down the center of the cell. And then the next row is saying what gets separated in anaphase. So in mitosis, you're separating sister chromatids. In meiosis 1, you're separating homologous chromosomes. And then meiosis 2, you're separating sister chromatids again. Okay, and then the last thing is what's the end result of the process. So the end result of mitosis is 2 daughter cells that are diploid, and that's if you start with the diploid cell. And then for meiosis 2, which is the end of meiosis, you get 4 daughter cells that are haploid, and that's again if you start with a diploid cell. Okay, so through this comparison, you can see that mitosis and meiosis 2 are a lot alike, and then a lot of the unique things that happen in meiosis happen during meiosis 1. Okay. Do you guys have any questions about the comparison or anything you want me to repeat if I went through it too quickly? Okay. So you can watch this animation on your own. It's just describing the features that are unique to meiosis when you compare it to mitosis. And this is another animation you can watch if you didn't understand the comparison of mitosis with meiosis. Okay. Okay. Let's see. So the first thing we're going to do is talk about polyploidy. And polyploidy is the situation of having extra sets of chromosomes. So let me rephrase that. Polyploidy is the situation of having more than 2 sets of chromosomes. Okay, so if your normal status is to have 2 sets of chromosomes, having a 3rd set, you would call that organism polyploid. Okay, so polyploidy is having an extra set, more than 2 sets of chromosomes. And you can produce a polyploid cell by an error in meiosis, and it's telling you that you could have a failure of the spindle to form properly, and then basically all the chromosomes go to one cell instead of being divided between 2 cells, and then you produce a cell that has more than 2 sets of chromosomes in it. So an error in meiosis can lead to a polyploid cell having extra chromosomes. It is important to note that most human polyploids fail to live past the very early stages of development, but in contrast to that, polyploidy is seen often in plants. So about 30% of flowering plant species are going to tolerate polyploidy well, and many crop plants are polyploids. Okay, polyploidy can be the normal condition of a plant, but in terms of humans, it would be an abnormal condition. And I don't think that we've seen it, so meaning that it's not viable. Okay, so in contrast to polyploidy, you can have something called aneuploidy, and we're going to get to that in a second. What we're going to talk about first is what can lead to aneuploidy. Okay, so aneuploidy is a slightly different condition from polyploidy. And first we're going to talk about non-disjunction, and the reason that we're talking about it is non-disjunction can lead to aneuploidy. Okay, so Non-disjunction we're going to focus on non-disjunction that occurs during meiosis, but non-disjunction can also occur in mitosis. Okay? And non-disjunction is simply a failure to either separate homologous chromosomes properly from each other, or a failure to separate sister chromatids from each other. Okay, so it can be a failure to separate a pair of homologous chromosomes from each other, or a failure to separate sister chromatids from each other. In either case, you're going to produce daughter cells that have an extra chromosome and daughter cells that are missing one chromosome. Okay, so on this slide, this is comparing a non-disjunction that occurs in meiosis 1 on the left with a non-disjunction that occurs during meiosis 2 on the right. So up here in meiosis 1, you're supposed to separate all of the pairs of homologous chromosomes. So all the homologous chromosomes are supposed to separate from each other. But here you have a non-disjunction where these two don't separate and both homologs go to the same pole. And then what you end up in the end is two daughter cells that have one extra chromosome and then two daughter cells that are missing a single chromosome. Okay, and then over here, this is a non-disjunction, what non-disjunction would look like in meiosis 2. So you're supposed to separate all the sister chromatids from each other. This pair of sister chromatids do not get separated, so they both go to the same pole. And then the end of the result is that for this cell over here, things are normal, so you produce two normal daughter cells. But for these two daughter cells, again, you're going to produce one daughter cell that has an extra chromosome and then one daughter cell that's missing a chromosome. Okay? Now, remember that the reason that we're focusing on non-disjunction and meiosis is because remember that these daughter cells give rise to your gametes. Okay? And so what can happen is if one of these cells is involved in a fertilization event, so let's say this cell is involved in a fertilization event where you have a cell that's n plus 1 fusing with a cell that's just n, then that can lead to an organism that has one extra chromosome, and that would be an aneuploid organism. Okay, so aneuploidy is the situation in which you have an aberrant number of chromosomes where you have one or a few extra or one or a few missing. Okay, so with aneuploidy, it's only one or a few extra or one or a few missing chromosomes. So in this case, we're not talking about sets like polyploidy. We're talking about additional extra chromosomes or missing individual chromosomes. Okay, so again, aneuploidy is going to be the situation in which you have either a few, one or a few extra chromosomes, or you're missing one or a few chromosomes. And this is just showing you in picture form what I just talked about, how non-disjunction during meiosis can lead to zygotes. And remember, the zygote is the cell that divides to give rise to the multicellular organism. How non-disjunction can lead to a zygote that has an abnormal number of chromosomes, and that's going to produce an aneuploid organism. Okay, and so aneuploidy in humans, this is showing you some of the viable ones. So there's actually in terms of ones we've actually seen in humans, we see in terms of having extra chromosomes, these are giving you examples of ones we actually see. So trisomy 21, trisomy 18, trisomy 13. So I'll describe what trisomy means in a second, but that basically means that they have 3 copies of chromosome 21. So the one extra chromosome they have a copy of is chromosome 21. And these are the ones that are viable in humans, but most trisomies are not viable in humans, and the same thing for most monosomies. Okay, so this is also, well, this would be a monosomy here. So most monosomies are also not viable or not seen in humans. Okay, so trisomy is aneuploidy where you have one extra chromosome. Okay, so trisomy, tri meaning 3, means you have one extra chromosome. So if you're trisomic, then you have one extra chromosome. If you are missing one chromosome, then you have monosomy, okay? And you would be described as a monosomic. Okay, so one example that you guys are probably familiar with is Down syndrome, or at least you're familiar with the syndrome. Down syndrome is usually caused by having an extra copy of chromosome 21. Okay, so another name for Down syndrome is trisomy 21 because it's often caused by having an extra copy of that chromosome. Okay, so again, you can think of monosomy and trisomy as two categories of aneuploidy. Okay, so monosomy and trisomy are two categories of aneuploidy. Monosomy is having a missing chromosome, whereas trisomy is having one extra chromosome. And the tri comes from the fact that if you have one extra chromosome, then there's one chromosome that you have three copies of, so that's where the tri comes from. And then the mono obviously comes from the fact that if you're missing one chromosome, then you have one chromosome where you only have one copy of it, and that's where the mono comes from. Okay, and then again, what can lead to these different conditions is non-disjunction. So non-disjunction can lead to producing aneuploidy in an organism. Okay. Do you guys have any questions about either polyploidy, non-disjunction, or aneuploidy?
So for those with genes of X, Y, are they still infertile? Like are all of the sex chromosomal?
I don't know about infertility with respect to the sex chromosome abnormalities. Sorry. But I can tell you that, well, obviously it's on there. And I discussed this earlier. Having a Y chromosome makes you male. So XXY and XYY people are males. And if you're XXX or XO, then you're female because you don't have a Y chromosome. Oh, that means it's missing the second sex chromosome. Yeah, sorry. So XO means that it's missing the second sex chromosome. And if I remember correctly, that's the only monosomy that we've seen, if I remember correctly. So meaning that we don't see monosomies for any of the other chromosomes in viable human embryos. Okay, so we take that to mean that it's not, we do not tolerate missing a chromosome very well. We also don't tolerate having an extra chromosome very well because there's very few trisomies that are also, there's very few trisomies that we see, and so we take that to mean that they're not viable. Okay, so let's do the lecture questions for this topic. Okay, so the first couple of questions just have to do with some of the introductory material that we had to talk about. So can someone tell me the relationship between germ cells and gametes? Right, and what process are germ cells going to have to undergo in order to produce gametes? Right, meiosis. So remember, germ cells in your body are diploid, and those diploid cells are going to have to undergo meiosis to produce haploid cells, and then those haploid cells develop into the gametes. So the relationship is that the germ cells give rise to the gametes. What is the relationship between gametes and a zygote?
Complementary gametes produce a zygote?
I wouldn't say complementary gametes. I don't know what that means.
Sperm and egg?
Yeah, so, and I would say fuse. So a sperm and an egg or two gametes are going to fuse, and that forms a single cell called a zygote. And remember, so the gametes are haploid, and when two of them fuse, it's going to form a diploid cell that you call a zygote. And again, that fusion process is called fertilization. Okay, so number 6, if a diploid cell goes through meiosis, how many daughter cells will be formed, and will the daughter cells be diploid or haploid? All right, 4 haploid. Number 9, when does synapsis and crossing over occur? Explain what occurs during crossing over. So when does synapsis and crossing over occur? Right, prophase 1, which happens in meiosis 1. And then remember, crossing over is the part that actually leads to genetic variation. Synapsis is just the pairing of homologous chromosomes. But once they've paired up, then crossing over can occur. So synapsis is why crossing over occurs frequently in prophase 1. of meiosis 1, and what happens again in crossing over? That's the product, yeah, but where were you going with that? Yeah, and basically by pieces you mean DNA. Yeah. So what's going to happen in crossing over is two homologous chromosomes. We described it as being two non-sister chromatids in the homologous pair are going to exchange DNA during crossing over. Okay, 11. Does independent assortment occur in meiosis 1 or meiosis 2? Right, meiosis 1, great. 14, what three things lead to genetic variation in sexually reproducing organisms? Not synapsis. Right, so the three are, independent assortment, crossing over, and random fertilization. And then remember, only two of those things happen during meiosis. That's being the crossing over and the independent assortment. Okay, so can someone, what I'm really asking in 16 is what's the difference between polyploidy and aneuploidy?
Polyploidy is the duplication of all of the genes, correct?
Not all the genes. I wouldn't say all the genes. Well, it is, but that's not the kind of the definition I would use for it. Right, having an extra set of chromosomes. And so just simply having an extra set or having more than two is going to make you polyploidy. And then aneuploidy is having what? Right, so one or a few more, or you're missing one or a few in aneuploidy. Okay. And then this we actually went through when we were talking about mitosis. So this is showing you, we actually went through both these, so I'm not going to go through them again, but I'll repeat what the answers were. So this is showing you a pair of homologous chromosomes, and the two long structures indicated by D here would be A, sister chromatids. And then 20 is asking what is E pointing to, and that would be C, the centromere. And those two questions we went through because they were in the mitosis one. Okay. Number 25, at the end of meiosis 1, which of the following statements would be true? Right, E. So 1, it's A, cells are haploid, and then also D, the homologous pairs are in separate cells. So remember, meiosis 1, you have the number of chromosomes, so the daughter cells are going to be haploid, and then also during meiosis 1, you separate these homologous chromosomes from each other, which means that each homolog in a pair is going to end up in a separate or different cell. Okay, so 26. Consider a diploid species where n equals 5. In this case, n is the number of chromosomes per set. Okay, so in this case, n is going to be the number of chromosomes per set. If an individual of this species was found to have 11 chromosomes, it would be categorized as a what? Right, E. So it's aneuploid because it has one extra chromosome. And if you had to be very specific, you would say it's specifically trisomic, because it's an aneuploid organism that has one extra chromosome. Okay, so 27, meiosis 1 produces blank, and meiosis 2 produces blank cells. B and C are the same, aren't they? Oh, no. It's C. My bad. So remember, you have the number of chromosomes in the first division. So the first meiosis 1 produces 2 haploid, and then meiosis 2 produces 4 haploid daughter cells. Okay. Number 30, homologous chromosomes are identical copies of each other. Yeah, that's B, false. So how would you describe, again, their difference? Right, they can carry different alleles for the same genes. And then I just want to, similar to the mitosis, so cell division lecture questions, I put flashcards in here for meiosis so you can go through and label the phases. And then if I describe something happening during that phase, make sure you put that on the back of that card. Are there any other questions that you guys see in this topic that you want to go through? Okay, so we're going to move on to the next topic then, which is DNA replication. And we're going to start DNA replication by doing a review, and you do need to know these things for this topic again, a review of DNA structure, because in order to understand how it's replicated, its structure plays a role in that replication process. So remember that inside of a cell, DNA is present as a DNA double helix. And so you have basically two strands of DNA, and the two strands of DNA are coiled around each other to form a double helix structure. So this is showing you the double helix structure with the two strands of DNA wound around each other. This is the two strands unwounded. Remember that each strand consists of nucleotides linked together to form that strand. So remember, for DNA, the monomer is a nucleotide, and then you link together many nucleotides to form a single strand of DNA, but a DNA double helix consists of two strands. Okay? Also remember that when you link together nucleotides, because of the way you link them, you're going to have one end of the strand that has that's called the 5' end, and the other that's the 3' end. And remember, the 5' end is the end with the free 5' phosphate group, and the 3' end is the end with the 3' hydroxyl group. And then in the DNA double helix, remember that the two strands are anti-parallel to each other, and that means that they run in opposite directions. So if this one runs 5' to 3' towards the ceiling, the opposing strand or the complementary strand runs 5' to 3' towards the floor. Okay, so their 5' and 3' ends are flipped and run anti-parallel to each other. And then also remember that there's specific base pairing that occurs within the DNA double helix in terms of the nitrogenous bases. So remember, what's holding the two strands together is hydrogen bonds that are occurring between the nitrogenous bases, and that's the pairing of the nitrogenous bases. And you get a very specific base pairing. So cytosine always pairs with guanine, so C always pairs with G, and adenine always pairs with thymine, so you'd say A always pairs with T. And so that's the specific base pairing that occurs during the DNA double helix. Okay, and again, when they're base pairing with each other, what's happening between the nitrogenous bases is you have hydrogen bonds between them. And again, that's what's holding the two strands together, are the hydrogen bonds that occur between the nitrogenous bases. Okay, so that's a review of DNA structure, and so now we're going to talk about how DNA replication takes place. And then just keep in mind that in a eukaryotic cell, this is what's happening during S phase. So when we went through the cell cycle and we said during S phase the DNA is replicated, and we said all the chromosomes are replicated, so this process that we're talking about now in a eukaryotic cell takes place during S phase. Okay. So before we knew how DNA was replicated, there were three hypotheses put forward for what could be happening, or three different ways that the DNA could be replicated. One was called conservative replication, the second was called semi-conservative replication, and the third was called dispersive replication. And experiments have shown that DNA is replicated semi-conservatively. Okay, so I'll explain each, but remember that how it happens in cells, regardless of whether you're a bacterial cell or a eukaryotic cell, is semi-conservative. So DNA is replicated in a semi-conservative fashion. Okay, so one hypothesis is that you had this. This would be conservative replication. So you start off with the parent DNA molecule. You separate the two parent strands from each other, which are blue. Then using the two parent strands, you synthesize a new daughter strand that's complementary to it. And then what happens is the two parent strands come back together, and then the two new daughter strands come together to form the new helix. So in this form of replication, you've conserved the original DNA double helix. So that's why it's called conservative. Okay, and so that's not what happens. This is semi-conservative, and remember, this is what happens. And so in semi-conservative replication, again, you separate the two parent strands from each other. Each parent strand is going to be used as a template to synthesize a complementary daughter strand, which is the new strand. But then the new daughter strand and the old parent strand are going to remain together. And so in this form, you've semi-conserved the old DNA double helix, because this helix here is half old and half new. So you've semi-conserved that old DNA molecule. Okay, and then dispersive was a third hypothesis. It seems unlikely. And what you have is you would have random, or not necessarily random, but you would have old and new kind of being intermixed with each other during the replication process. And that would be dispersive replication. Okay, but again, this is the way that DNA is going to be replicated in a semi-conservative manner. Okay, and so one thing that I alluded to when describing semi-conservative replication, and this is important in terms of understanding DNA replication, is that both strands in the DNA double helix are going to serve as a template for the synthesis of a new daughter strand. Okay, and this is just to contrast it. When the next thing we're going to talk about in gene expression is transcription. And in transcription, only one of the strands of DNA serves as the template. Okay. But in DNA replication, both strands are going to serve as a template. And the way that that works is if you have your original DNA double helix, ATGCACG, I'll just make it short. So this would be the other strand. Okay, so one thing that's obviously going to happen during DNA replication is that you're going to have to separate the two strands. And that's because so they can serve as a template, so that exposes the nitrogenous bases. So this is separating the two strands. So this strand goes up here, and then this strand goes down here. And then once they're separated, then they can both serve as templates. So this one serves as a template for the new daughter strand. So an enzyme is going to come in, and because there's a T here, in the new daughter strand that's building, it's going to put, because there's an A here, it's going to put a T. And then, so you're just using the base pairing rules, and that's how it's used as a template. So the sequence of nucleotides in this parent strand is going to determine the sequence of nucleotides in the new daughter strand, and you're just using the base pairing rule. So then connected to that, you would put an A, then a C, then a G, then a T, then a G, then a C. And the same thing's going to happen down here. This strand is also going to be used as a template. So you put an A, T, G, C, A, G, G. And again, both strands are going to be used as a template during this process, and that's how they're used as a template. So the sequence of nucleotides in both parent strands is going to determine the sequence of nucleotides in each of the new daughter strands that you're building. Okay. So DNA replication is going to involve a lot of different proteins because it's a rather complicated process. And so the top part are the different enzymes that are going to be needed. And these aren't the only one, we're leaving one out. But the four of the enzymes that you're going to need are DNA polymerase, DNA helicase, DNA primase, and DNA ligase. And in terms of what they do, I'll write down the gist of what they do here, and then we'll describe them in more detail. But DNA polymerase synthesizes the new daughter DNA strands. And that's a simple way of saying that this is the enzyme that actually connects together the nucleotides to form the new daughter strand. So the way that it synthesizes the new daughter strand is by connecting together the nucleotides together to form a daughter strand. Okay, DNA helicase is the enzyme that's going to separate the two strands of DNA from each other during DNA replication. So we said that the two parent strands have to be separated from each other. That's going to be done by DNA helicase. Okay, and then a third enzyme you're going to need is DNA primase. This is not, if I asked you to define this on an exam, this is not what I would be looking for. So the short version is that it builds a primer, but that's not telling me much. You need to tell me what a primer is when you're defining it on an exam. So if I ask you to define it, and I'll specifically state that on the exam, that if I ask you for DNA primase, you need to tell me specifically what the primer is. So it builds this thing called a primer. And so what I'm going to do here in parentheses is write down what the definition of a primer is. So a primer is a short strand of nucleotides. Okay, so basically you take nucleotides together, you connect them together, but you only connect together a few. And so it's a short strand of nucleotides. In DNA replication, DNA primase makes an RNA primer, which means that the primer you build is made out of RNA nucleotides. Okay, so initially you're going to have these short strands of RNA within your DNA molecule. So another thing that's going to have to happen during DNA replication is that you're going to have to remove those primers and replace them with DNA. Okay, I'll state the reason why you need primase now, and it's important that you know this. So the reason that you need primase is that DNA polymerase cannot start a new strand on its own. Okay, so DNA polymerase cannot start a new strand on its own. It can only build off a pre-existing strand. Okay. Okay, and then you need DNA ligase. And this, we'll just simply say for right now, it's not going to make much sense, connects together adjacent fragments of DNA. Okay, so DNA ligase connects together adjacent fragments of DNA. And that's going to be its role in DNA replication. Okay, and then two other things that you're going to need during DNA replication are these things called deoxyribonucleoside triphosphates. And these are the nucleotides that get connected together to form the strand of DNA. So we said that nucleotides are the monomers. These are the monomers that actually get added together to form the strand of DNA. So you kind of already know what these things are, but DATP, so we talked about what ATP is. DATP is the same as ATP except that the sugar is deoxyribose. Okay, so DATP is deoxyribose, adenine as its nitrogenous base, and then 3 phosphate groups. Okay, so DCTP would be the same thing, except its nitrogenous base would be cytosine. DGTP, the nitrogenous base is guanine. And DTTP, the nitrogenous base is thymine. So when you're using the parent strand as a template and there's a thymine in the parent strand, then this nucleotide with an A is going to come in because it base pairs with a thymine, and then DNA polymerase is going to connect that to whatever nucleotide comes before it. And what we're going to see is that what happens during the addition of a new nucleotide is the last 2 phosphate groups get lopped off, which is why in a strand of DNA you only have one phosphate group per nucleotide. Okay, and so you need the deoxyribonucleoside triphosphates. Another thing that we'll mention, we'll have special slides for it. So these are the monomers. These are the building blocks for the new strands, the new daughter strands that you're building. This is also where the energy comes from to build the new daughter strands. Okay, so obviously building a new strand of DNA is going to require energy, and the energy comes from each of these deoxyribonucleoside triphosphates when they come in to be added. And then the last thing that we're going to have to talk about that you need during DNA replication are these things called single-strand binding proteins. And so one of the things that we told you has to happen during DNA replication is that you have to separate the two strands from each other, which is what DNA helicase does. But what happens is when you separate them, you don't, when you do DNA replication, you don't actually separate the two strands completely from each other. You just separate a small portion of the total chromosome from each other. So you're going to have this single-stranded region, and then here's where it's still double-stranded. And then what you're going to do is continually unwind or separate the DNA in this direction. And so this would be a double-stranded region, and these would be where they're separated, and you have two single-stranded regions. The single-strand binding proteins bind to these single-stranded regions, and what they do is they prevent the two strands from coming back together. Okay, so most important, so they're called single-strand binding proteins because they bind to single-stranded regions, but their role in DNA replication is that they prevent the separated strands from coming back together, and that's going to allow them to be used as templates during the replication process. Okay. And this is basically what we just went through. This is the table taken from your book describing the importance of the enzymes that we just discussed, and then also the single-strand binding protein. One we're not going to discuss, so you can cross it off the list so that you're not confused, is topoisomerase. We're not going to talk about its role in DNA replication. And then the one protein, or not, they're not proteins, sorry, the one thing that's missing that we just talked about would be the deoxyribonucleoside triphosphates. Okay. So you have to start at a certain place within a chromosome when you replicate the DNA, and that is called the origin of replication. So DNA replication is going to begin at what is called the origin of replication. And so that's where you're going to begin to separate the two strands and begin the replication process. And what happens is, again, you're not going to separate the two strands completely from each other. You're only going to start in a small region, and then that region is going to expand during DNA replication. So you can see that here is the origin of replication for this chromosome. So that's where you start to separate it. So I'll just draw it larger over here. So the origin is where the two strands start to be separated from each other, and the DNA replication process is going to begin. And then what happens is you start at this origin of replication, and then replication proceeds in both directions from that origin of replication. So you're going to replicate in this direction, and you're also going to replicate in the opposite direction. And so we say that DNA replication is bidirectional. Okay, so at the very top of the slide it says it's bidirectional replication, and that's what that means, that it proceeds in two directions away from the origin of replication. Okay. Also, you can see, so this is showing you that replication is going to proceed in both directions, and then they've drawn the arrow in here because there's more space. And then also, when you separate the strands like this, you're going to produce two Y-shaped regions, and those Y-shaped regions are called replication forks. So at an origin of replication, you're going to produce two replication forks. And that replication fork, that Y-shaped region is going to move, because what happens is, as this, you're going to separate the two strands from each other in this direction, and as you separate the two strands, the Y-shaped region is going to move. And so this replication fork moves to the right, whereas this replication fork moves to the left. Okay, so it's important that you know that you start at an origin of replication, that DNA replication is bidirectional, and that when you separate the two strands from each other like that, you're going to produce these regions called replication forks. And at an origin of replication, you're going to end up with two replication forks that are going to move in opposite directions. Okay, the next 2 points that I want to make are just comparing the typical prokaryotic chromosome with the typical eukaryotic chromosome. And so this is showing you replication of a bacterial chromosome, which is a prokaryotic chromosome. And so typically remember that this chromosome is circular in these organisms. And then also typically you have a single origin of replication. So that means you have a single start site. Okay, in contrast to that, so what's going to happen is you're going to start at the single origin of replication and then just move around the circle until the two replication forks meet at the other end. But for a linear chromosome in eukaryotic organisms, what you typically have is not one origin of replication, but having multiple origins of replication. What do you think having multiple start points does?
Speeds up the process.
Right, speeds up the process. And eukaryotic chromosomes are typically larger than prokaryotic chromosomes, so speeding it up is important. So having multiple origins of replication is going to speed up how fast you replicate that chromosome. And also these chromosomes are linear. Okay, so remember eukaryotic chromosomes are typically linear, and then they're going to have multiple origins. They can have hundreds or even thousands of origins of replication, so hundreds or even thousands of starting points. Okay, and so this is repeating some things that I've already said, but it's nice because it shows you a picture. So again, this is showing you where the two strands have been separated from each other. Here, this is showing you an enzyme we're not going to talk about, which is DNA topoisomerase. This is helicase, and this is 1/2 of the origin of replication. So in this case, this helicase is moving to the left. And so this Y-shaped region here is one replication fork. Okay, so that's where the two strands are separated, and you have a Y-shaped region. So here's the Y-shaped region. And then once you separate the two strands, and again, that's performed by DNA helicase, these single-strand binding proteins are going to bind to the single-stranded regions, and that prevents the two strands from coming back together. Okay, so you need to separate the two strands. Then the single-strand binding proteins come in and prevent the two strands from coming back together. This is going to allow them to serve as a template for the synthesis of a new daughter strand. But remember that the enzyme that actually builds the new daughter strand is called DNA polymerase, and that cannot start a new strand on its own. And again, what that means is, let's say that this is, I'm just going to draw one DNA template strand, so A, T, G, C, A, C, G, A, C. And let's say this is where you start. DNA polymerase can't just come in and add a T here, because that would be starting a new strand. Okay, so what has to happen is DNA primase is going to have to come in and lay down this short stretch of RNA that we call a primer. And because it's RNA, remember you use uracil instead of thymine, so that U, A, C, G, and then a U. And then once you lay down this short primer, then this provides something for DNA polymerase to build off of. So once you've laid down this short sequence of nucleotides, then DNA polymerase is able to build off of that primer. Okay, so again, you need the primer because DNA polymerase cannot start a new strand on its own. So once you've separated the two strands, and the single-stranded binding proteins have come in and prevent the strands from coming back together, DNA primase is going to have to come in and lay down primers that are complementary to both parent strands. Okay, and so DNA primase is going to make this short RNA primer, and also the primer is going to be laid down so that the primer is anti-parallel to the strand that it's complementary to. Okay, so meaning that if the 5' end of the template strand is down here, that means the 3' end of the primer is here, and here's the 5'. Okay. So again, you need primase because DNA polymerase can't start a new strand on its own. Once primase builds the primer, then DNA polymerase can come in and synthesize off that primer. Okay, and that's going to form the new strand of DNA. Now there's two things about DNA polymerase that you need to know, one of which you already know, which is it can't start a new strand on its own. The second thing is that DNA polymerase can only build a new strand in one direction. It only builds new strands in the 5' to 3' direction. And what that means, to say this a different way, is that DNA polymerase can only add on to the 3' end. Okay, so if it's given something to build off of, it can only build off the 3' end of what's available. It never adds on to the 5'. So because it always adds on to the 3', the direction of synthesis of the new daughter strands is going to be 5' to 3'. Okay. So this is just showing you, and essentially what DNA polymerase is going to do, again, is it's going to link together nucleotides to form the new strand of DNA that you're making. And so again, if this is the template strand, because there's G here, a deoxyribonucleoside triphosphate that has cytosine as its nitrogenous base is going to be the next nucleotide added in. And DNA polymerase is going to connect this nucleotide to this nucleotide. And then it moves on down the template strand. And then what happens essentially is when this gets added into the new strand, these two phosphate groups get lopped off as a group, and those two phosphate groups bound together is called pyrophosphate. So those get lopped off, and then the pyrophosphate gets split into two phosphate groups. Okay? And the reason that I mentioned those two reactions is both of those reactions are exergonic. So when you release pyrophosphate, that releases energy, and then when you split pyrophosphate, that also releases energy, and that's where the energy is coming from in terms of synthesizing the new DNA daughter strand. So I told you that synthesizing the new DNA strand comes from these deoxyribonucleoside triphosphates, and that's because lopping off pyrophosphate is exergonic, and then splitting pyrophosphate is also exergonic, so both of those reactions release energy, and that energy is used to incorporate that new nucleotide. And that's all this is trying to show you in this case. Actually, don't ignore this slide. That's not what it's trying to show you. Ignore this slide. I'm not going to go through this. Okay, so DNA polymerase is the enzyme that's building the new strand. DNA ligase, we said, is going to be needed in order to connect adjacent fragments of DNA. What's shown here that we haven't explained yet is that because the two DNA strands that make up the parent DNA molecule are anti-parallel, and because DNA polymerase can only add on to 3' end, we're going to see that the two new daughter strands are built in different ways. One's going to be synthesized continuously, and the other, which means you're going to have one long fragment, whereas you have to build the other one as a series of fragments. Okay? Regardless of which one you're talking about, you're going to have to lay down a primer or primers for them. So what they're showing you here is that they've laid down a primer in blue, and then you build off of it. Red is the DNA. Okay? And then you laid down a second primer here. Red is building the new DNA off that primer. So eventually what's going to happen is you're going to remember the primers are made out of RNA, so you're going to have to remove them. And so what happens is a different type of polymerase comes in and removes the RNA, and then it fills in this gap with nucleotides. But what happens is it can add the last nucleotide here, but it can't then connect this last nucleotide to the first nucleotide of this fragment. It can't make that covalent bond. Okay? So it can fill in the gap with all the nucleotides that you need, but it can't connect the last nucleotide of this fragment with the first nucleotide of the following fragment. And that's what DNA ligase does. DNA ligase comes in and specifically forms that covalent bond and joins those two fragments of DNA together. Okay, and DNA ligase is going to be needed on both strands. On the lagging strand, DNA ligase is going to be needed more than once. So I haven't described. On the strand that's built in fragments, you're going to need DNA ligase more than once. And on the strand that's built as one fragment, you're only going to need DNA ligase once. Okay, so the next thing we're going to talk about is how DNA replication occurs at a replication fork. And when we discuss this, we're going to call the strand that's built in fragments the lagging strand, and then the strand that's built as one continuous strand, the leading strand. Okay, so I'm only going to draw 1/2 of the replication fork. This is 1/2 of a replication fork, and so this is going to be, in this case, moving in this direction. In order to understand which one's going to be the leading and the lagging, I have to label the ends for you. So this is, let's say this is the 5' end up here, and this is the 3' end of that strand. So that means the 3' end for this strand is down here, and then the 5' end is over here. Okay. So obviously this is where you've separated the two strands, and then the single-stranded binding proteins would be bound here, preventing the two strands from coming back together. And helicase is moving in this direction, separating the two strands. Okay, so remember, once you've separated the two strands, then DNA primase can come in and lay down the primer, and you're going to have to lay down a primer on both strands. Okay, so if you lay down a primer here, I told you that the primer is laid down so that it's anti-parallel, and it's also synthesized in the 5' to 3' direction. So when you lay down this primer, here's the 3' end of that primer, and here's the 5' end. But for the primer laid down here, here's the 5' end, and here's the 3' end. Okay. Now, once you've laid down the primer, then DNA polymerase can build off that primer, but remember, it can only add on to a 3' end, which means for the bottom strand here, DNA polymerase comes in, builds off this strand in the direction that the replication fork is moving, and then you can just keep on going as the two strands are separated. So this strand is going to be synthesized as one long continuous strand, and that's the leading strand. Okay, at the top here, because you can only add on to the 3' end, you're going to build off this primer, but you're going to build away from the direction that the replication fork is moving, which means that as you separate the two strands, you're going to have to continually lay down a primer, build away from it. Then as you separate it more, you lay down another primer, build away from it, and you have to build this strand as a series of fragments, and that's going to be the lagging strand. Okay, and then what you're going to have to do eventually during the DNA replication process is you're going to have to remove all the primers that you laid down, and so you'd have to remove this one primer down here, and then you'd have to remove every primer that you laid down for the lagging strand. And then remember that once you remove that this primer, here's the 3' end and here's the 5' end. And so this would be the 3' end of this previous fragment. So once you've removed the primer, then you can build off this 3' and fill in the gap with nucleotides. But then once you add the last nucleotide in here, you can't connect this last nucleotide with the first nucleotide of that fragment. And again, that's what DNA ligase is for. DNA ligase comes in and will connect those two adjacent fragments of DNA, and then that seals the new daughter strand. Okay. And so in terms of being able to differentiate between what's going to be the leading strand and what's going to be the lagging strand, because it depends on how I label them, to determine which ones it's going to be, all you have to do is lay down a primer. And if you have to build towards the replication fork, that's going to be the leading strand. If you lay down a primer and you have to build away, that's going to be the lagging strand. Okay. So in terms of being able to distinguish the two on a test, when you lay down the primer, if you have to build towards, that's going to be the leading strand. If you build away, then that's going to be the lagging strand. Okay. So this is going to describe DNA replication in E. coli. And there's actually, so for this process, you'll have to know the roles of two DNA polymerases. We also have multiple DNA polymerases that play different roles. The two that appear to play the major roles in E. coli are DNA polymerase 1 and DNA polymerase 3. Okay? And so you also have multiple DNA polymerases. And then there's other DNA polymerases that play a role in DNA repair. the two, in terms of E. coli, that play a major role in DNA replication are going to be DNA polymerase 1 and DNA polymerase 3. Okay, so DNA polymerase 3 is the polymerase that I'll just say builds off the primers. So every time there's a primer laid down and you synthesize a new daughter strand off that primer, it's DNA polymerase 3. Okay, and then in E. coli, ours are not labeled 1 through 3. Ours are labeled alpha, beta, gamma, delta, etc. So DNA polymerase 1, and again this is in E. coli, this is the enzyme that removes the primers. And replaces them with DNA. So it synthesizes that replacement strand of DNA. Okay, so basically we're just going to go through the same process that we just went through, but now distinguishing between what DNA polymerase 3 is doing, what DNA polymerase 1 is doing. And so in this picture, they're also not showing you helicase, but it's moving in this direction in this case. So in the picture, the two strands are being separated, moving in the left direction. And so again, the first thing that's going to have to happen is once you've separated the two strands, primase is going to have to lay down a primer. Once you lay down the primer, DNA polymerase 3 can come in and build off that primer. In this picture, DNA polymerase is going to come in and build towards the direction that the replication fork is moving. So the top strand is going to be your leading strand, but then at the bottom, DNA polymerase 3 is going to have to come in and build away from the direction that the replication fork is moving. So that's going to be your lagging strand. So at the bottom, you're going to have to, as you separate the two strands, you're going to have to continually lay down primers and build away from the replication fork. But each time, it's going to be DNA polymerase 3 that comes in and builds off each of the primers that's laid down. Then eventually what's going to have to happen is DNA polymerase 1. They've already shown this happening. There was a primer here. DNA polymerase 1 is going to remove a primer, replace it with blue DNA, and then what happens is a DNA polymerase 1 can fill in all the nucleotides that you need, but again, it can't take this last nucleotide of this fragment and connect it to the first nucleotide of this fragment. And again, that's what DNA ligase does. DNA ligase will come in and form that missing covalent bond in the new strand of DNA. Okay, so basically it's the same thing we just went through, but being more specific in terms of the polymerases that are needed in terms of building off the primers, and then the polymerase needed to remove the primers and replace it with DNA. And again, this is happening in E. coli, which is a bacteria. And the reason that we went through the specifics here is because this is where we understand the process the most, because we initially studied DNA replication in E. coli, and then later we're able to apply that to other organisms. So that's why we know so much about it. Okay, and so what we're going to do is we're going to stop and we'll take a 10-minute break there, here, and then we'll continue on and finish up DNA replication and probably finish the questions and also start gene expression today.